In this video, we'll have a look at systemic gas exchange. Now, let's first start with that phrase. When I'm saying systemic, in this case, I mean everywhere except the alveoli. So any other gas exchange happening in muscles or your liver or your heart, etc., would be considered systemic. You'll also see that there is a diagram that looks very familiar, looks almost identical to this, and that's about alveolar gas exchange. And if you look carefully, the only difference between the two diagrams, it comes down to the direction of the arrows. So let's have a look at systemic gas exchange. Now, what we have to do is we have to get basically CO2 out of a tissue. It's being the result of aerobic respiration. And I have to get CO2 and I have to move it into the blood for transport to the alveoli so I can breathe it out. Now, what we're going to do is look at the three main mechanisms that CO2 uses to travel between, say, an active muscle and the alveoli via the blood. And let's start with the most simple, and that is where CO2 enters the blood and remains as a dissolved gas. Now, remember, things go from high concentration to lower concentration. So that's the main factor that's going to be driving this movement of gases from systemic tissue into the blood. So this is the most straightforward one, where CO2 just crosses and it dissolves as CO2 gas within the blood itself. Now, this only represents a very small amount. We're only looking at about 7%. Now, let's go to the next way. CO2 can remain as CO2, but what it does is it basically hitches a ride. It binds itself onto a macromolecule for transport. In many ways, it's like it dials up an Uber, hops in, takes a lift to the alveoli. So it still remains as CO2, and this makes up about 23% of the transport of CO2. And in some cases, it binds onto plasma proteins. You know, that's basically its Uber. It hops on, binds onto the plasma proteins, catches a lift to the alveoli where it's going to be released. Or the other option is it comes along and it binds onto the hemoglobin. That's what HB stands for, hemoglobin. Now, in both cases, CO2 remains as CO2. So up to now, we've seen that CO2 can be a dissolved gas. That's only about 7%. It can catch a lift either on plasma proteins or on hemoglobin. But in all three of these cases so far, it remains as CO2 as it heads off to the alveoli in the lungs. But this still only represents about 30% of the CO2. Most of the CO2, about 70%, gets involved in this equation. Now, this equation isn't as bad as it looks. It's fairly straightforward. Let's walk through it. Remember, CO2 goes from high concentration to an area of low concentration. And what happens is CO2 within the red blood cells interacts with water. Of course, there's water there. It's abundant. And it uses an enzyme called carbonic anhydrase, and it forms this acid. Now, this acid then breaks apart. It dissociates into bicarbonate, that's the HCO3, and also into one hydrogen. Now, when you look at this, you notice that, well, here we had CO2, and then CO2 doesn't seem to show up here. But of course, if you know your basic chemistry, you know that CO2 is actually hidden here. The carbon and the two oxygens are hidden within the bicarbonate. Now, it's really important to look at the ratio here, that for every bicarbonate, I get one hydrogen. So we're looking at basically a 1-to-1 relationship, that for every bicarbonate, I have one bicarbonate, I end up with one hydrogen. And we'll come back to that in a second. Now, the bicarbonate, I don't want it to build up, because as I'm bringing in more CO2, I'll be making more bicarbonate and more hydrogen. I don't want these things to build up and linger in the cell. If they do, The problem is this is a reversible reaction, and of course, if I end up with high concentrations of these two products, they now become reactants and the equation may reverse. It's that classic sort of mass part of the equation. So what happens is the bicarbonate is transported out of the red blood cell. Now the type of transporter is an antiport. And remember from our first course that the deal with an antiport is that when one thing moves out, something must move in. So it's a two-way opposite movement. So in this case, bicarbonate is moved out by the transport system and chloride is moved in. Now why chloride? It's just the way the system works. That's the way this transporter works. A bicarbonate goes out and a chloride comes in. There's nothing magic about it other than that's the way it works. Now we're going to give this process right here, the movement of the bicarbonate out and the chloride in as a chloride shift. Why? Because I'm just shifting chloride in order to accomplish what I need to. So that was one of the first great things about this chloride shift is I'm moving bicarbonate out so I don't end up with a buildup here and either slow down the reaction or, in a worst case scenario, reverse it in systemic tissue. That wouldn't be good. The other great thing is that when I move bicarbonate out, the bicarbonate in the blood acts as a buffer. And this is great because, of course, we want buffers in our blood. We don't want our blood pH to wander all over the map because, of course, macromolecules, especially proteins, are liable to denature. So this acts as one of the parts of the buffering system of the blood. So I get a 2-for-1 deal. All right. So, and remember, basically CO2 is hidden in there, right? 1 carbon and 2 oxygen are hidden in there. Let's go back to the hydrogen. Now, let's remind ourselves, for every bicarbonate I made 1 hydrogen. And the hydrogen basically can come along and it can go up to oxygenated hemoglobin. That's what that HbO2 means. It's basically saying it's hemoglobin carrying oxygen. And what happens is this hemoglobin, when it interacts with hydrogen, basically it releases the oxygen off of here. So all of a sudden I start to end up with a buildup of oxygen that's now free oxygen that's now being released. And of course it's going to go from high to low and move in that direction. All right, so there's a couple other points I want to make here. Is if you really follow this equation, that the more CO2 I load in to a red blood cell, and that's often what we call it, loading. When I'm putting something on a red blood cell, I've loaded it in, okay? That the more CO2 that I load in, the more oxygen that'll drop off. So this is a really good way to match basically tissue needs. So if a muscle tissue is very active, it's going to be making a lot of CO2, and that, via this equation, leads to the release of more O2. So I can match demands within a tissue by this simple equation. The next thing that I want to point out is to go back to this relationship of bicarbonate and hydrogen ions. So for every bicarbonate that exists, there is 1 hydrogen. And we're going to see in this chapter, and also in the heart chapter, we're going to make reference to it also, that one way the body can get a sense as to how much carbon dioxide there is by looking at the number of hydrogen ions. Or another way of saying that is by looking at the acidity of fluids. Because if you consider this, that for every one of these, I make one of these. So if I know how many of these exist, it gives me a good idea how many of these exist. And of course, what's the thing about this is that this is where the carbon dioxide is hidden. So just to remind you, this is about systemic gas exchange. What happens at alveolar gas exchange? It's actually the opposite. When you go and into your notes, into the book, and you look at the diagram for alveolar gas exchange, you'll see it's exactly the same. The only difference is all the arrows are reversed because all of these are reversible processes. So make sure you spend some time, walk your way through this. This is the essential components of systemic gas exchange.